The aim of this procedure is to assess the impact of a progressive colonization on the host hepatic metabolism. This is accomplished by first progressively colonizing germ-free mice. The colonization process is monitored by evaluating the urinary excretion of microbial co-metabolites through NMR spectroscopy. Subsequent to monitoring of the colonization process, a liver biopsy is collected from the mouse and prepared for metabolic profiling. The hepatic metabolic profile can then be acquired from the intact biopsy using non-destructive high-resolution magic angle spinning NMR spectroscopy. Ultimately, this use of proton NMR spectroscopy reveals various metabolites involved in metabolic pathways such as energy and oxidative stress pathways.
So this experiment can provide insight into the liver functions during colonization. It can also be applied to other systems such as kidney.
To colonize the animals, remove germ-free mice from isolators and house them in a conventional husbandry room in cages equipped with a filter in front of the conventional animals, which serve as controls. Then, mix half of a 3-day-old litter taken from the control conventional cage with the litter of the germ-free animals. Collect urine in a 1.5 ml microtube by handling the mouse over the tube and help micturition by gently massaging the bowel. A minimum of 20 microliters is required for acquisition with a 5 mm NMR probe, but it is recommended to use 30 microliters to improve the quality of metabolic profiling. Snap freeze the urine immediately in liquid nitrogen.